Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 49, where I will talk about phenylpropanoids, benzenoids, coumarins and tannins. This is basically I will give you an overview in this class. So, these are the concepts which are to be covered, and let's go to the slide. So, as I have mentioned before also that this is basically the phenyl ring and the hydroxy group which constitute the phenolics compound. Phenolics must fulfill this criteria, but also I mentioned that it is not always mandatory, even the cinnamic acids and then phenylethanol, these are also coming under phenolics because it's originate from phenylalanine. Okay, and some of the function of phenylpropanoids I have mentioned in the previous class. So, this one there is no point of discussing again, and I ended the class with this schematic diagram, where I said that hydroxycinnamic acid basically the P, and these are different P1, P2, P3, P4, P5, say P6, so many, so on, so on. So, that means that these are maybe different transformation which includes hydroxylation, methylation, glucosylation, chain cleavage, so many. Okay. And okay. So, this is again an overview which I have given in the previous class. So, now come to here. So, this is called phenylpropanoid and phenylpropanoid acetate skeletons. So, phenylpropanoid skeletons I have mentioned that which is basically C6C3, and phenylpropanoid acetate skeleton 3 into C2 and C6C3 this formed. So, and this contributes this one is this acetate skeleton is coming from the this malonyl acetyl derivatives, and eventually it forms different phenolic compounds. Okay. Like the flavonoids, where it requires this like anthocyanins. So, there the role of this acetate skeleton is important, and this is again coniferyl alcohol, which is a major constituent of the lignin components. So, which is also called monolignol, and this is a typical example of quercetin. As I said that this is basically flavonoid. So, this is coming here, whereas in case of coniferyl alcohol, which is where there is no involvement of malonyl derivatives. So, there is no this orange structure is there. So, acetate origin is not required there. So, now let us make an overview. So, what I have said that cyclimate pathway contributes to phenylalanine or tyrosine or both. So, the phenylalanine are C6C3 compounds. I have shown the structure of phenylalanine, and then phenylalanine by the action of the enzyme phenylalanine ammonia lyase, it converted into transenamic acid. So, this is phenylalanine ammonia lyase which converts phenylalanine into transenamic acid, whereas the tyrosine this I draw in a slightly different way, but it is all same. So, tyrosine converted into 4-coumaric acid, this is called 4-hydroxy cinnamic acid or 4-coumaric acid, or even also it is called para-coumaric acid. This is ortho, this is meta, this is para. So, the enzyme here is called TAL, phenylalanine ammonia lyase tyrosine ammonia lyase. So, which also released a molecule of ammonia and makes paracumaric acid. So, this TAL exists mostly in the monocot plants, because monocot plants are basically dominated by coumaric acids, particularly the grasses. So, their TAL exists, it is possible to measure the TAL from there. Okay. And aminoxy phenylpropanoic acid. So, this is basically act as an inhibitor. So, scientists they have studied the pathway by feeding this. So, which basically act as an inhibitor of this PAL. Okay. Now, go back to the structure. So, this is PAL. PAL makes either cinnamic acid or cinnamic acid, and this cinnamic acid converted into subsequently into other simple phenolics, phenylpropanoids or hydroxycinnamates. So, which are also C6C3 compounds. Okay. And as I also said that from tyrosine 4-coumaric acid, which is another hydroxycinnamic acid, can be formed by the action of TAL, but that is mostly confined to the graminea grass families. Okay. Next, these simple cinnamic acids will be converted into the other hydroxycinnamic acids, that is cinnamic acid converted into paracoumaric acid, caffeic acid, ferulic acid, all these things. So, these are all C6C3 structures. I will show you, and now this C6C3 structure, what will happen? This will contribute towards the formation of lignin. As I have mentioned, this is the lignin pathway, and which is C6C3, simple C6C3 is monolignol, but when that joins in multiple numbers which form the polymer, that is why this N is given here N. So, that is why it is called lignin, whereas two such molecules form lignans. Okay. Now, this C6C3 structure joins with malonyl CoA, either 3 molecules of malonyl CoA or 2 molecules of malonyl CoA, and it makes different structures which are again C6C3 C6 structure. So, this is chalcone. Chalcone is basically the precursor of anthocyanin, and the enzyme is called chalcone synthase. So, chalcone synthase requires, what is the substrate of chalcone synthase? One is malonyl CoA, 3 molecules of malonyl CoA and 1 molecule of paracoumaryl CoA. So, the paracoumaryl CoA plus 3 into malonyl CoA makes chalcone, or we call tetra hydroxychalcone. We will see this in details later. The enzyme is called CHS. Similarly, malonyl CoA joins with coumaryl CoA and it makes another structure which is called stilbene. So, stilbene and chalcone subsequently converted into flavonoids, so which are which constitute different classes like anthocyanins, flavonones, isoflavones, flavones, so many things. Also, C6C3 structure joins together and form polymeric structure like suberin cutin, which basically deposits in the epidermal cell layer and which protects the plant from different abiotic and biotic stresses. And again this C6C3 C6 structure which I have shown here, this contributes to the formation of condensed tannins. So, where multiple of these units joins together and form this condensed tannin structure. So, you see here the C6C3 is the backbone, from their different addition or substitution or chain shortening leads to the formation of different compounds. Chain shortening is C6C1. When we talk about C6C1, then that is the example of chain shortening, like C6 C1, like benzenoids, these are the chain shortened compounds. So, I have mentioned this again in the previous class. I mentioned this earlier in the previous class. What is the chain shortening here? Yeah, this is hydroxybenzoids here, and all other things I have already mentioned. So, let us now see. So, formation of major phenolic groups from phenylalanine and the core phenylpropanoid structure pathway. So, phenylalanine converted into trans-cinnamic acid by the action of the enzyme PAL. This is PAL, and that trans-cinnamic acid contributes directly towards formation of benzenoid compounds. That is why benzoic acid derivatives is written here, and trans-cinnamic acid by the action of one hydroxylase, it forms this 4-hydroxycinnamic acid, which is also called paracumaric acid. I have drawn the structure just before. So, this is a hydroxylation added at the 4th position. That means, this is 1, 2, 3, 4, 5, 6. Okay. And then, so this paracumaric acid contributes to the formation of caffeic acid, ferulic acid and other important phenylpropanoid structure. And this paracumaric acid also undergoes activation. This is called activation of hydroxycinnamic acids, where CoA molecule joins. So, and by the action of an enzyme called coumaryl CoA ligase or 4-hydroxycinnamoyl CoA ligase. So, ligase means addition of CoA molecule, which requires ATP and MgCl2 to perform its function, and it forms the paracoumaryl CoA. So, paracoumaryl CoA is a very active molecule, because it takes part into different components such as lignin precursors, such as stilbenes, such as coumarins, and of course, for the flavonoids and anthocyanins. So, paracoumaryl CoA is a very, it is called activated hydroxycinnamates, because a CoA molecule is added, and then upon joining with malonyl CoA, I have mentioned just in the previous slide, it forms the chalcone, and from chalcone it forms different flavonoid structures. So, so this is the structure of hydroxycinnamic acid. So, this is the trans-cinnamic acid, which converted into by the action of C4H, cinnamate 4 hydroxylase, which makes a 4 position OH added. So, this structure is called 4-hydroxycinnamic acid or paracumaric acid or 4-coumaric acid, all are correct. And then we will also see, ideally from 4-coumaric acid, if what will happen, first a hydroxyl will add here, and in so that is the job of the hydroxylase, and the next job will be that this will be replaced by methyl group. So, and eventually this structure is this one, which is called ferulic acid, or you can say 4-hydroxy 3-methoxy cinnamic acid, 4-hydroxy 3-methoxy cinnamic acid, or it is called ferulic acid. Although the formation of ferulic acid is not straightforward in this way, which we will see later, but theoretically ferulic acid can be formed from paracumaric acid by enzymatic steps. First there will be hydroxylation, then there will be methyl transferase. So, where which requires a methyl group donor like SAM, and which makes ferulic acid, and ferulic acid subsequently again undergo hydroxylation at 5 position. This is 4 position, this is 5 position, this is 3 position. So, and then this is for hydroxylase, and then there will be that will be methylated. So, methoxy eventually. So, this is called synapic acid, SINAPIC synapic acid, or 4-hydroxy 3,5-dimethoxycinnamic acid, or 4-hydroxy 3,5-dimethoxy cinnamic acid. These are the different names. Now, hydroxycinnamyl alcohols, this we will discuss again when we will talk about the monolignol biosynthesis in next class, but briefly paracumaric acid is converted into paracumaryl CoA. This is activation of hydroxycinnamide, then what will be happen? There will be a reductase activity. So, this enzyme is called 4-Cl, this enzyme is called reductase CCR, cinnamyl CoA reductase, and leads to the formation of this structure. This is cinnamaldehyde or coumaraldehyde, coumaraldehyde, and then coumaraldehyde again there will be the action of CAD, alcohol dehydrogenase, and which eventually form the coumaryl alcohol or paracoumaryl alcohol also you can say, and this constitutes one of the units of the lignin monomer. So, this is called monolignol. Similarly, from ferulic acid another monolignol structure originates, which is coniferyl alcohol. From synapic acid another monolignol structure originates, which is called synapyl alcohol. So, detail of this I will talk in the subsequent class. Now, coumarins. Coumarins are also important molecules. So, coumarins basically they act as a defense compound, and they basically formed from the coumaric acid is the basic structure. And then it forms for example, coumaric acid, then coumaric acid ultimately it forms umbelliferone, which is 7-hydroxycoumarins, and then you will get a compound called soralin, which is not good for health, because if you touch in the skin it forms blisters, and coumaric acid converted into ferulic acid, and subsequently the ferulic acid by the action of ferulate 6' hydroxylase. So, at the 6' hydroxylase, so 1, 2, 3, 4, 5, 6 at this position 6' hydroxylase, it eventually forms this structure scopolamine. So, this is one example of hydroxycoumarin. So, here is the structure of soralin, which is coming through the umbelliferone. Okay. So, and this is the general skeleton of the coumarin structure, and a linear furanocoumarin, which is 8-methoxysoralin, synthesizes human skin to UVA light. So, this compound present in this plant and cause severe blistering on skin, followed by exposure to UV irradiation. So, this is furanocoumarin, and you see the blisters. Now, come to the condensed tannins. Okay. Condensed tannins basically are formed by joining of this C6, C3, C6 in multiple numbers. Okay. The first one is called the where n equal to 1 to 10, when n equal to 1 to 10. So, it also contains 3 hydroxy group. So, this is basically the structure of the condensed tannins, and the hydroxy group of a hexose are esterified with the gallic acid, and here the bottom one is the hydrolyzable tannins, which is basically originate from the gallic acid. And yeah, so this is condensed tannins you find in different plants like Horitokis, Bahera, these plants you will find condensed tannins. So, this is again another example of this hydrolyzable tannins, which is found in the chestnut, Castalagin, this is the name of this. And one example of this I have put it. Okay. So, this is again gallic acid. So, this is basically the glucogallin is the ester form of gallic acid, and this finally forms this gallic acid, this condensed tannins. Now, biosynthesis of benzenoids, as I mentioned that benzenoids are C6C1 compounds. So, basically it forms from C6C3 structure by a chain cleavage. So, cleavage means that it basically cleaves out the C2. So, there are the phenylalanine which makes transenamic acid, as I have mentioned, by the action of the PAL, and then by 4Cl it makes cinnamyl CoA. And what happens, the transenamic acid can take part in the formation of benzaldehyde. When you talk about the benzaldehyde, if you see this structure, this is C6C1 structure, this is C1, right. Okay. So, what happens from transenamic acid, it can directly form benzaldehyde, which is called CoA independent non beta oxidative route. CoA independent. So, that means there is no involvement of CoA here, and this is the non beta oxidative or non oxidative route, which what happens that there will be the formation of one intermediate, which is called beta hydroxy phenylpropanoic acid, and there will be maybe the enzyme is called benzaldehyde synthase. This mostly is unstable compounds, and eventually it makes benzaldehyde, and benzaldehyde contributes to the benzoic acid. So, this is one route. So, similar route also exists for hydroxy benzoates molecule as well. We will discuss sometime later, and another. So, this is one route. The route number 2 is basically CoA dependent beta oxidative route, where cinnamyl CoA forms beta hydroxy phenylpropanoyl CoA, and then beta oxophenyl propanoyl CoA, and then it forms the benzoyl CoA. So, here it involves different enzymes. For example, phenyl beta hydroxy phenylpropanoyl CoA. So, that from cinnamyl CoA, this is 4Cl, there is another enzyme called CNL. So, CNL is that basically a specific enzyme which contributes towards the formation of this benzenoid structure. Here there is called cinnamyl CoA ligase, but this is termed as CNL. So, normally the 4Cl is used for activation of hydroxycinnamates. When there is no hydroxy, only cinnamate. So, there CNL terminology is used, and it forms the 3-hydroxy this 3-phenylpropanoyl CoA, and then it makes beta oxophenyl propanoyl CoA, and then finally it makes benzoyl CoA. So, benzoyl CoA when it forms, so there is an enzyme called ketoacyl CoA thiolase, this one, this is called KAT. KAT is a recently discovered enzyme, ketoacyl 3-ketoacyl CoA thiolase, THIOLASE. Thiolase means basically the removal of the it is a thioester is acted, removal of the CoA thioester, and that leads to the formation of the benzoyl CoA, and benzoyl CoA subsequently forms the benzoic acid, and then this benzoic acid may contribute towards the formation of salicylic acid, because if this is one position, this is 2. So, if I have hydroxy added, then it becomes salicylic acid. So, long back one work from Rutgers University, they mentioned about this Iller-Askin's group, but later it was found that that may be existed, but mostly the salicylic acid is formed from chorismic acid by isochorismate synthase, which makes isochorismic acid, then there will be isochorismate pyruvate lyase, which basically release the pyruvic acid and produces the salicylic acid. And this is the most acceptable route of salicylic acid. So, salicylate is also a benzenoid because this is C6C1 structure. So, vanillin is also C6C1 structure. So, what we see here that the core phenylpropanoid pathway which contributes towards the formation of coumaryl CoA and malonyl CoA, and by the action of chalcone synthase it forms chalcone, and subsequently the pathway contributes towards the flavonol. The pathway also contributes towards the anthocyanin. The pathway also contributes towards the unusual pigment fluvapen. Maybe I will discuss all these things in due course, and flavones, and also that these chalcones can take part in the formation of isoflavones, which are basically dominant in the legumes. So, this is basically the pathways and branch points leading to the formation of major flavonoid groups and related compounds like isoflavones, isoflavonoids, anthocyanins, and then proanthocyanidin, that is condensed tannins, which also form from this way. So, like this joins together, and also stilbenes, stilbene we will see now. So, stilbene and chalcone. So, what happens, there is an enzyme called stilbene synthase, okay, which requires again 3 molecules of malonyl CoA plus coumaryl CoA, and which what will happen, it liberates 4 CoA thioesters and carbon dioxide, leading to the formation of a stilbene structure which is called resveratrol. Resveratrol is a strong antioxidant, and it basically accumulates in the red grapes. Therefore, consuming red wine is good because it contains resveratrol, which gives protection to heart diseases. But resveratrol is produced grape basically, it is act as a defense compound, particularly it gives protection against fungal diseases. Okay. In another complex structure of this stilbene is viniferin, which is also found in this. So, these basically act as an antifungal compound. So, later this structure has been synthesized and it is used as a fungicide, but originally it is basically found in on Vitis species. The same malonyl CoA and coumaryl CoA also contributes towards the formation of chalcone by the action of chalcone synthase. So, this is the structure of tetrahydroxychalcone. As you see, this red one is basically coming from the acetate unit, okay, acetate malonyl unit, and whereas this one is coming from the coumaryl unit, the black one. And chalcone is the precursor of various flavonoids like I have mentioned flavonones, flavones, isoflavones, flavonols, anthocyanins, and this is typically the structure of flavanone and flavon 3 ols and flavones. Maybe I will in one of the classes I will draw this structure to explain the differences between these. Whatever required, I will not go to that much complex way, but whatever required I will discuss this. So, isoflavones, as I mentioned, this is found in the legumes, and genisticin one such isoflavones, and these are all dietary phenolic compounds. So, this is very important for health purpose. And then similarly, this malonyl CoA and this paracoumaryl CoA or malonyl CoA with cinnamyl CoA or malonyl CoA with caffeoyl CoA, that means malonyl CoA joins with cinnamyl CoA or it joins with paracoumaryl CoA or even it joins with caffeoyl CoA and forms different structure like benzyl acetones, like aryl pyrrone. So, one good example is xylotinin. So, xylotinin is found in the primitive pteridophyte xylotum. So, you see that it originates from this pathway, and I have mentioned about xylotinin and when I talk about the evolution of these metabolic pathways. Maybe again I will discuss that when I talk about the xanthones. and so this is come under aryl pyrrone. Then similarly, stylopyrones are produced. Histidine is one example, and chalcones, of course, stilbenes are example. And the example of the stilbenes, one is the resveratrol, another is pinosylvene. Pinosylvene is another example of stilbene, chalcone, naringenin, eridictol. So, these are all examples. So, this is basically the designation of 1x, 2x, 3x refers to the number of molar equivalent units of malonyl CoA, when whatever number is required. For example, in formation of styropyrone require 2 molecule of malonyl CoA, whereas cylotinine or aryl pyrrone require 1 molecule, whereas stilbene and chalcone requires 3 molecules of malonyl CoA. So, this is what it is about. So, and then this eventually forms different complex flavonoid subclasses. So, this all looks very Greek. So, I will not really discuss in details, but the example you must know a little bit. Diazenin is one isoflavonoid, which is there in legumes. Similarly, medicarpine, which is phytoalexin sort of compound. So, that gives protection to plant against pathogen attack. Naringenin contributes to the formation of anthocyanin. This is an audone. Audone is also contributing to the pigments, like in canna species. So, that the pigment is basically originates from the audone. So, these are the some of the examples of different flavonoid classes. So, with this I end this class. So, I have given you a brief overview of the phenylpropanoids, benzenoids, condensed tannins, and stilbenes, and little bit of flavonoids here. So, I hope you understand and you have enjoyed. So, see you again in the next class. Thank you very much.